what they call axon terminals, boutons, some people call it. We'll just let's call it a neuron for right now. Okay? Right here at the level of the muscle, there's going to be some sort of connection. We call it a neuromuscular connection. And that's where we're going to get into more bigger picture. The neuromuscular junction right here, we're going to look at bigger picture. So here's where we have it. Same thing. So again, this is a neuron coming down, and an electrical impulse is going to be coming down that axon, coming down that neuron. So we want to move our skeletal muscle. Our brain sends an electrical impulse down the neuron to the muscle cell. That's where we are. Okay? So the electrical impulse comes down, and when it reaches the end of the neuron, it causes these vesicles, right here in green, to fuse with the plasma membrane. That leads to what they call exocytosis. Remember what that is? So this fusion of the vesicles is going to release that neurotransmitter into this pocket or cleft right here. This is important because if you notice very carefully, this neuron is not physically connected with the muscle. There's a gap there. That means that electrical charge ends right there. The only way the electrical charge can be transmitted to the muscle, that stimulus, is via that neurotransmitter. That's it. Okay? That neurotransmitter gets released and goes into what we call the synaptic cleft. Then we have receptors for that neurotransmitter. So specifically what the neurotransmitter is acetylcholine. We're going to be talking about, there's probably 20 or 30 different neurotransmitters. Acetylcholine is the only one I want you to know by heart. So know acetylcholine. Okay? Acetylcholine is what's inside these vesicles. And again, it gets released. What's going to happen? I'll give you the big picture first. These neurotransmitters will get released in this pocket right here. They're going to bind to this receptor right here. These receptors are the acetylcholine receptor. They bind acetylcholine. And the best way to think about that is imagine this is a receptor. And we have a nice pocket for that to bind to. So this is the acetylcholine receptor. Okay? And this is acetylcholine. So acetylcholine will get released and it binds to that receptor. So this is in the plasma membrane. Okay? Now if you remember, receptors can do different things. Some of them are channels, and this is where we're going to go. There are two types of channels we're going to be talking about today. One of them is called, in this case, receptor mediated. And the other one is called, well, let's see, one of them is mechanically gated, sorry. The other one's electrically gated. Let's talk about what that means. When acetylcholine binds, what's going to happen? Well, it's going to open up a pore. There's going to be a channel that opens up. So here's the plasma membrane. Here we have all kinds of sodium just waiting on the outside. And all kinds of potassium waiting on the inside, because we just talked about we have that big gradient. And now all of a sudden, what do? We open up a giant big pore. And now what happens? The sodium flushes in to the cell like crazy. So acetylcholine binds. That causes the channel to open. That leads first to sodium to coming in. So what's going to happen? Let's go over here. We start with negative 70 millivolts. All of a sudden, we're bringing in tons of positively charged ions. All over the place. I mean, look at how many acetylcholine receptors there are. This entire region of the muscle cell is going to be saturated with all this incoming sodium ions. That's going to change that negative charge here to positive. And that's what's going to lead to the increase in the voltage. Can you see how that can happen? Increase the amount of positive ions and the voltage goes up. Later on, what you're going to see is when it reaches a certain climax, a certain level, that channel is going to change. And now it's not going to allow sodium to come in. it's now going to allow potassium to come out. So all of a sudden, at this point, at this voltage, it shuts off sodium and says, okay, potassium, go out of the cell. And now all of a sudden, the opposite thing happens. So first sodium, then potassium, and as a result of that, you get this big spike in voltage. At a certain point, the channel closes up again. It doesn't allow either sodium or potassium to come in or out. And then what happens is the sodium-potassium pump, it's always going, builds up the gradient again, and again we have 10 times and 10 times again. So in a very short period of time, I mean we're talking microseconds, milliseconds, okay, very small amount of time, right at one area of the plasma membrane, we have this huge spike, comes back down, and then goes back to normal. Very, very fast. And that's what's happening right here. So we're causing this rapid increase in sodium coming in, a rapid efflux of potassium coming out, and then going back to baseline. What's going to happen? Well, think about the other type of channel we have. We have something called a voltage-gated channel. Okay, I have better pictures than this, let me just explain it right now. Voltage-gated. That means, imagine, right here, okay, or right here. This part of the plasma membrane, notice there's no acetylcholine receptors. But there are voltage-gated channels. So what does that mean? Well, anytime the resting membrane potential is perturbed, is disrupted, it's now going to change, and it's going to open up. It's going to open up in response to a change in voltage. So you can imagine if right here, this is my muscle cell again, and right here is where the neuromuscular junction is. They call it the motor end plate. That's the part of the muscle cell. Okay? Right here is that motor end plate of the muscle, and right here I'm binding acetylcholine receptors, and that's leading to that rapid increase in sodium inside the cell. Right? That area of the cell is going to become highly positively charged. And then as that sodium starts to diffuse down the cell, that's going to change the voltage along different areas of the cell, isn't it? And what's that going to do? Well, that means these channels right here are going to open up and more sodium is going to pour in, which is going to lead to more diffusion. More sodium, more sodium, more sodium, more sodium, more sodium, and as a result that voltage travels all the way down the entire length of the muscle cell. Shortly after the potassium comes in, the sodium comes in, the potassium level comes out. So it's a combination of this mechanically gated channel and then the propagation of this voltage gated channel. This leads to that voltage going all the way down the entire length of the muscle cell. Am I going too fast? I'm just giving you the big picture. Okay? So let's look here at the neuromuscular junction. So again, this is where the neuron synapses to or connects to the muscle. Now I said it's not a physical connection, so don't keep that in mind. It's not a physical connection. There's a synaptic cleft there. And again, you can see that here. Okay? Don't worry about that. Okay, so here is the release of that neurotransmitter. So again, you have the charge comes down, that leads to the release of acetylcholine, that leads to binding to the receptor. You can see right here, here's acetylcholine receptors. They're going to bind acetylcholine. You can see the kind of bluish, greenish binding to that receptor. That's a nice picture of that receptor. Again, it binds acetylcholine. Right now it's closed. Doesn't allow anything through it. But then all of a sudden, when it binds that, it now allows sodium and potassium to go in and out. Now this picture is a little bit confusing because in reality, it allows sodium first, then potassium. It's in response to voltage. So keep that in mind, like I just explained here. Okay? But it does allow voltage to go through. Then you have voltage-gated channels. So here you have another channel that responds to voltage. So this guy allows all the sodium to come in. And then over here, once that sodium level comes over here, the charge changes, it opens up that channel as well. Okay. Oh, thank you.
Did you have done before, right?
I have.
Okay, thanks for your help.
Does it have to be done today?
You can get it done by next Monday.
I can
I
try.
won't tell.
Thank you.
See you later.
Okay. Okay. So that makes sense in general so far. So we have the binding, it opens up the channel. That opening of the channel leads to that voltage changing, and the change in voltage leads to other channels opening. And again, you can imagine it's a domino rally. As long as they keep opening, it's going to allow more sodium to come in, and that's going to cause more and more receptors to open up, and that change in voltage is going to propagate or travel down the entire length of that muscle cell. Are we okay? I know it's hard, kind of a hard concept to visualize. So here's another way of visualizing it. Again, this is not in your book, but another picture that I found somewhere, and it just kind of shows you this. So what we're starting off here is we're starting with the acetylcholine binding and causing the first initial increase in voltage. So the sodium's already come in, and now what we're looking at is simply the voltage-gated channels. So this is that muscle cell. What's going to happen all along the sarcolemma, the plasma membrane. So here's where it starts off. It's negative on the inside, positive on the outside, but all of a sudden, we change the voltage. It's more positive now. That causes the channel here to open up. That leads to more sodium. That causes the channel here to open up. Here, here, here. And all of a sudden, you can see how that propagates. We go from being negative to positive, and then back to negative again. And so you see this change in voltage that goes all the way down the entire sarcolemma, plasma membrane. Okay? It's one way to visualize that. Where is it going to go? Well, if you remember, the T tubules, or the transverse tubules, are continuous with the plasma membrane. So when that voltage travels down the sarcolemma, every single time it finds a T tubule, it's going to go down right here. And that electrical current is going to go down here. It's that electric current that causes the sarcoplasmic reticulum to release calcium. Okay? There are sensors, for lack of a better word for right now, they call them photoreceptors, that bind right here, that basically sense the change in voltage. And when they sense a change in voltage, they open up and release calcium into the cytoplasm. So they're just waiting for the voltage to change. And that voltage changing causes calcium to be released. So you can see that right here. That calcium that was originally sequestered or built up and maintained here inside the sarcoplasmic reticulum is then allowed to dissipate into the cytoplasm. So the cytoplasm is going to increase the concentration of calcium. If you remember, we're going to see it in just a few minutes, the troponin complex, which senses calcium, is going to pick that up and move the troponin. Okay? Let's look. Tropomyosin. So here is the troponin complex right here. It senses calcium. And all of a sudden, when calcium levels go up, it's going to change in shape. And you can see now that changing the shape moves this tropomyosin molecule and exposes the active sites. Can you see now the holes that I didn't show you before? So now the active site can be bound by myosin. It's just waiting to find an active site. It finally can bind to it. And now it can bind to it and pull. And that's what contracts the muscle. Okay? I know it's a hard concept. I want you to be able to understand this all the way back from the let's say, the neuron, and be able to describe it all the way to this point. Okay? That's a hard concept to understand, but I want you to understand it at that level. Okay? So this just shows you how this basically works. And I'll show you the big picture here because I don't expect you to know this kind of detail. Okay? This is, in general, how this works. As I said before, we have the myosin, and it exists like this, and we have to cock the heads. Okay? That cocking of the head, this is the high energy state, the cocking of the head requires energy. That's ATP. So you have to put ATP and use ATP to cock the head. Once it's cocked, all it needs to do is bind that active site and it'll pull, 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 pull. But it has to get cocked to do that. And that's what they're showing you right here. Okay? So you have to cock the heads by using ATP. And then it binds and forms what's called a cross bridge. Not a term I'll ever ask you, but all a cross bridge is where myosin and actin bind together. They form a cross bridge. And ATP is required again to cock the heads. Does that make sense in general? Okay. That's a fun little tangent to this, in case you're wondering. I wouldn't expect you guys to know this, but when the body dies, the ability to make ATP is one of the first things to go. Okay? ATP is required to go from this to this. So the muscle is in a contracted state shortly after death because there's no ATP. What do you think we call that?
Rigor.
Rigor. That's why you have rigor. Rigor mortis.
Is that why when a person dies, they always move everywhere?
Well, they lose muscle control.
Right.
Yeah, right. Yep. Yep. Okay? And then shortly after death, a couple hours after death, then that starts to break down, enzymes break down, muscles break down, and you lose rigor. But within 4 to 6 hours of death, you get rigor mortis due to that. So this shows you a diagram of basically what's happening. So again, you have those channels. I drew it very crudely on the board. We're going to get to more of this in chapter 11. I'm not going to focus a lot on this today. I think this is more complicated than we need to. Chapter 11 we'll get to after the next test. But again, your resting membrane potential is negative 70. You're going to have the sodium channels open, and now that voltage, which is on the y-axis, is going to go up and up and up until it reaches this point. Sodium channels will close, potassium channels open, 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 open. It overshoots, we'll talk more about that later. It goes too far, and then it has to recover and come back to the resting membrane potential. And again, the thing to keep in mind is the sodium-potassium pump is always working. So you're disrupting that gradient, but the pump will bring it right back as soon as it can. So very shortly after that voltage, it'll recover. So right now, I have a voltage right here. It'll change, but it goes very, very quickly, and then it comes back to resting membrane potential. It's very short-lived. So it's like a little spike, 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 and it just goes back to resting all the way down. This is better, there's probably better pictures online or videos online that might explain it better than I can do it verbally. This shows you something not in your book, but in case you're curious about exactly how the troponin complex works. Again, troponin, which is right here, senses calcium levels. So it's going to sense how much calcium is in the cytoplasm. In a resting muscle cell, in other words, not contracting, the calcium levels are low. And so the troponin complex exists like this, and it separates actin from the myosin. Remember, myosin only wants to bind to that active site. It can't, because that is being blocked. What happens here is the calcium levels go from 2 to 4. See, 2 to 4, and as a result, it causes the troponin to move. That causes the tropomyosin, which is this little doodad right here, to move away, and now the active sites are exposed, and you get muscle contraction. So that's the sensing by calcium. Troponin, tropomyosin. You should know that just because of one thing. When we talk about smooth muscle, there's a difference there. So troponin and tropomyosin. I think I talked about everything that's on page 9. Make sure I don't forget anything. How does it end? Well, we know that muscle contraction doesn't last forever. Muscle contractions are short-lived, unless we have continuous stimuli. So if, for example, that neuron is constantly firing, sending nerve impulses down here, boom, 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 we'll continue to contract our muscles. However, if it's a single nerve impulse that comes down here, it's going to lead to what we just described, and to muscle contraction, but it's going to be very short-lived. Why? Well, the neurotransmitter, the acetylcholine, is going to diffuse out into the environment. It's normal diffusion. There's also an enzyme called acetylcholinesterase, I don't remember what it's called, that breaks down acetylcholine, and again, reduces the amount of neurotransmitter that's there. So as a result, what's going to happen, at least at this level, is the stimulus will be reduced and eventually go away. Unless, again, you get another impulse, and then again, more neurotransmitter will be released. But with just one, you get a short little burst and it ends. Also, calcium levels start to get sequestered back in the sarcoplasmic reticulum. So yes, it releases it quickly, but then it starts to grab it back up again. Okay? unless it receives a second signal. Then it'll release it again, and again, and again. But if the stimulus ends, the calcium levels go back down because they go back to the sarcoplasmic reticulum. And as a result of that, when the calcium levels go back down, the troponin complex and the tropomyosin go back to their normal orientation of blocking the active sites on actin. And therefore the contraction ends. So the only way to have continuous contraction is to have continuous stimulus. Does that make sense? Because that's what we're going to do after the break. Let's talk about that in more detail. What we just talked about is one of the hardest things in anatomy. This is at least in 101. It is. Okay? Really 2 lectures, and this first part is hard. The second part is not so hard after break. It's not as hard as we just talked about. So, do you guys have any questions about this in general before we go to break? Stacey?
No, not yet.
Not yet. You will. No, I'm just kidding.
Yeah, no, I will. I'll make a list as I go back
It's
through
one of those
everything.
things, honestly, I'll be right with you, Cody. It is one of those things that it probably would be good for you guys to look online, look for videos, find something that might visually help you understand this. There's some available online, I know that, for example, okay? Because sometimes just looking at a 2-dimensional picture that's static, not moving, is hard to see. And I can't do it with the time we have allotted in the class. So work on it, try to understand it. I would expect you guys to understand that. And I'm not going to have you write an essay and describe this in detail. But I do want you to understand some basic ideas, okay? It starts with a nerve impulse. It leads to acetylcholine release. That leads to a change in the membrane potential. That leads to, again, traveling down the sarcolemma. It leads to the T-tubules, which release calcium. That's a big trigger. How do muscles get contracted? Calcium. They go up, right? Calcium leads to troponin changing, moves tropomyosin. That allows the active sites on actin to bind to the myosin. The myosin grabs and pulls. That brings the sarcomeres closer together, which brings the whole muscle shorter. I know I just went really fast. If you guys can describe that, understand that idea, you'll be fine in this part. If you can't, you've got some work to do. Okay? And I know you probably don't get it all right now. A hard concept to understand. Yeah, Cody.
So where does the acetylcholine vesicles actually being released from the axon?
It's actually, yeah, so the end of the axon, there's these little, they call knobs, or boutons if you're French. That's what these are right here. This is the end of the axon. So right here at the end of the axon, those vesicles are located in. So basically when the electrical impulse comes down, it causes exocytosis of acetylcholine. And acetylcholine is the only one, as I said, we need to know right now. And that's the only one that is responsible for skeletal muscle. So every skeletal muscle in your body is activated by acetylcholine and non-acetylcholine. Other neurotransmitters do other things. But for skeletal muscle, that's all we need to know. Anything else? You laugh. Okay. Let's see, 6:46. Let's come back at 6:51.